everyone. Today I would like to start our lectures on plant cell and tissue. So a typical plant body contains the shoot system and the root system. As we can see in the pictures, the root system contains anything that usually you can see above the ground. So it has many functions including photosynthesis, reproductions and dispersal, food and water conductions. So while in the root system usually is anything that under the ground that you cannot see. So it's anchor the plants in the soil, absorb water and nutrients and conduct water and nutrients, and of course it also serve as the food storage. Example is like a carrot plant. So what is the differences between plant growth versus to animal growth? So the key differences lies on the determinate cell growth. So in animal, after the fertilizations occur, the zygote cell will start to divide undifferentiated. However, after a certain critical stage, the cell differentiated and formed tissue. So from this point onward, the developmental of the cell is sealed. For example, if your cell is to develop into the corneal cell, it will not going to develop into different or other cell types. So the skin cell will develop into the skin cell type and will not later on develop into different types. This is the meaning of the determinate growth. So of course there are some exceptions of this system, such as the stem cell in bone marrow. So most animals have a pre-programmed body plans. For example, you have 10 fingers, toes, 10 toes, 2 eyes, and hearts with 4 chambers. So it will be the same for everyone except due to the mutations or accidents. So in plant cell, they contain the indeterminate growth. What is called by indeterminate growth? So these plants will develop, the plant development is continued by embryogenesis cell called Meristem cell. This meristemic tissue is undifferentiated cell and they do not have pre-programmed body plans. So this meristemic cell tissue can later on develop into, for example, shoot system, root system, stem system, or even leaf. So they are constant like leaf shape and branching pattern that is you follow according to different types of plant or different species, but you can never predict where a new branchings will come out from a tree. So plants have tremendous developmental plasticity. So when you cut or the plant is wounded, it can grow and regenerate by new meristem cell. So and the last thing that the plant cell quite different from the animal cell is have a cell wall. So this is of course they have different in depth characteristic, but these two characteristics is very important that I will explain later on. By using this knowledge, you can use it for application in plant biotechnology. So let's look at the common between plant cell and animal cell. So firstly, the fertilizations of haploid egg cell and haploid sperm nucleus in plant and animal cell is similar. Then later on, the diploid cell will start to divide and regulations of the balance between cell growth and cell division will help create the shape of the embryo. So and later on, the formation of distinctive pattern of organ, cell, and tissue differentiation will generate different types of cell that's suitable for different function. Plants have tremendous developmental plasticity. So what is means by plasticity? The loss of plant part can regenerate by meristem cell. Even more, the single cell plant can generate into the entire plant. So these plant cells are called totipotents, which means that if we provide the stimulus, for example hormone, plant growth regulator, it can initiate the plant to grow into a different cell type. In the plant tissue culture, we always use the same application. We provide certain types of enzyme so that the plants can grow. For example, if we interested to grow only the root system, so we provide the stimulus, and in the plant tissue culture system, so we can produce the roots in the box conditions. So in plant tissue culture, the callus formation of callus, which is the unorganized parenchyma cell, is actually very useful for genetic modification, and it is normally initiated under the sterile laboratory conditions, which means if the cell contain virus free, if the cell is virus free, then it is useful for genetic modification purposes. Because plants cannot escape predator and other kind of damage, but they can readily repair wound and reconnect the vascular stent by differentiating the appropriate cell types. So plasticity enable the change form and shape and functions of the plant cell towards respond to the different environmental change. So this involves a lot of the cell-cell communications is important in plant development, but cell reconnection is likely less important than it is in animals, since plant cells keep the same neighbor cell throughout their life. So for the continuous week, we will discuss on the whole of plant life cycle, starting from seedling, mature plant, flower, and fruiting mechanism. So let's look at type of the cell or tissue that build up a plant. So we have three types of the plant cell: parenchyma, that storage or responsible for photosynthesis, such as those in mesophyll cell; storage of starch in endosperm, or cotyledon in primary cell wall. And then we have parenchyma cell, which work as a glue to support new growth regions of the plant. So we can find this cell in growing shoot and leaf. The third types of the cell is parenchyma cell, which is more rigid for support. Example is the fiber and sclerite. So when look at this table, it shows you the locations of tissue system. So all of the different types of the cell will be later on become the building block for a different tissue system. So we have, if you make cross sections of the stem cell, you can see that the tissue system divided into three system. That is dermal tissue system, which is the outer part, ground tissue system at the middles, and the inside inner system is the vascular tissue system. As we all know, the vascular tissue system contains the xylem and phloem that normally and very important to transport water and mineral as to provide food for other plant part. So the ground tissue system is responsible for energy transformations, photosynthesis, food storage, and regeneration and support. So they contain a lot of the chlorophyll here in the ground tissue system, while the outer layer, the dermal tissue system, contains a cell wall that given the protections and prevent the water loss to maintain the water content of the plant. So talking about the cell wall, look at the pictures here. This is the structures of the cell wall. A cell wall is a structure layer surrounding some types of cell. So it is just outside the cell membrane. It can be tough, flexible, and sometimes very rigid. It provides the cell within both structural support and productions, and also act as filtering mechanism. Cell walls are present in many prokaryotes, in algae, fungi, eukaryotes, including plant, but are absent in animal. A major function is to act as pressure vessel, preventing overexpansions of the cell when water enter. So the compositions of cell wall varies between species and may depend on cell types and developmental stages. So the primary cell wall of the plant is composed of polysaccharide, cellulose, hemicellulose, and the pectin. Often other polymer such as lignin, suberin, or cuttins are anchored or to be embedded in plant cell wall. So here I want to highlight a particular important structures here, that is cellulose. Cellulose is an organic compound consisting of a linear chains of several hundreds to many thousand of the beta 1 to 4 linked D-glucose unit. So it's make the cellulose an important structures component of the primary cell wall of green plant. So because plant can use the photosynthesis to absorb carbon dioxide and to change it into oxygen and water, this carbon dioxide, the carbon in the environment, can change to become the carbon in the cellulose fiber. So then it's undoubtedly to make the cellulose is the most abundant organic polymer on earth. So for example, cellulose content in carbon fiber is about 90%, and the wood is 40 to 50%. So this large amount of the cellulose is actually normally used to produce the paperboard and paper, and the rest of the biomass residue can be converted to become the renewable biofuel. So we know that the hydrolysis of the cellulose can produce sugars, and the sugar after fermentation can produce the bioethanol. However, the hydrolysis of the cellulose is always hindered by the large amount of lignin. So lignin will act as a protection wall to prevent any chemical enter to the cell wall to digest the cellulose. So Here I share research done by my group members to use lactase that immobilize into a nanoparticle to digest the lignin and hence improve the hydrolysis of corn curve. So in this paper we show that after we expose the corn curve biomass to the magnetic lactate immobilized nanoparticle that produced in our lab, we managed to digest the lignin to almost 40%, and due to the efficiency of the lignin digestions, we managed to convert the cellulose about 23% higher compared to the non-lactase digestions method. So this will improve the productions of bioethanol for renewable energies. So this is one of the examples I want to show you guys on how the knowledge of this plant developmental biology can help for the development of some biotech products. So let's continue to the other types of the plant cell. Plant are multicellular eukaryotes with tissue system made of various cell types that carry out specific function. As I explained just now, plant tissue system fall into one of two general types, that is meristematic tissues and permanent cell tissue. So cells of meristematic tissue are found in meristem cell, which are plant regions of continuous cell divisions and growth. Meristemic tissue cells are either undifferentiated or incompletely differentiated, and they continue to divide and contribute to the growth of the plant. In contrast, permanent tissue consists of plant cells that are no longer actively divided. Therefore, in plant tissue culture laboratory, we are using only the meristemic cell tissue to generate a new plantlet. So meristemic tissue consists of three types based on their locations in the plant. Apical meristem contain meristemic tissues located at the tips of the stem and roots, which enable a plant to extend in length. Lateral meristem facilitate growth in thickness or give in a maturing plant. Intercalary meristem occur only in monocot, as the base of the leaf bladder and the nodes. This area where leaf attach is to a stem. This tissue enable the monocot leaf bladder to increase in length from the leaf base. For example, it allows lounge grass leaf to elongate even after repeated mowing. Meristem produce cells that quickly differentiates or specialize and become permanent tissue. Such cells taken on specific row and lost their ability to divide further. They differentiated into three main types that I explained just now: dormant tissue, vascular, and the ground tissue. So this slide shows the cross sections of the terminal buds where it's located the lateral meristem, shoot apical meristem, and the root apical meristem. So in laboratory, these three parts will be used to work on micropropagation. Here is an example of my another study working on the micropropagations of orchid from shoot meristem. So basically we take the shoot meristem and we sterile them in the laboratory conditions. After sterile, we grow them on the defined media contain only the sterile defined media. So this is the callus that produce under the tissue culture conditions, and if you can see that the cell concentration increasing from day 1 to day 7 after exposures to the different types of stimulus. So like I mentioned just now, so this is how the callus look like, as I mentioned in the previous slide. So the callus is a growing mass of unorganized plant parenchymal cell. In living plant, actually, callus cells are those cells that cover a plant wound. In biological research and biotechnology, callus formation is induced from plant tissue sample or explant after you cut out the meristemic cell, and surface sterilizations and plating onto tissue culture medium. So the medium that we use to produce this orchid callus is supplemented with plant growth regulator, which is also known as a hormone, such as auxin, cytokinin, and ghrelin, to initiate calcium callus formations, or also known as somatic embryogenesis. Somatic embryogenesis means that the most of the genetic makeup are similar. So the use of the callus cell. So why we are generating callus in tissue culture? So callus cell is actually not necessarily genetically homogeneous, because a callus is often made from structural tissue, not the individual cell. But callus cells are often considered similar enough for standard scientific analysis. So because it is a clone for the same cell. For example, an experimenter may have half a callus undergo a treatment as the experimental group, while the other half undergo a similar but non-active treatment as the control group. So means that the two types of the callus is also callus from the same mother plant, but is undergo different types of the stimulus. So as I mentioned just now, plant callus derived from many different cell types can differentiate into the whole plant. That the process called regeneration through the additions or stimulus. So for the whole plant, each single cell have a copy of transgenes in every cell. So the gene of interest then can be inserted into the callus using a lot of the techniques such as ballistic bombardment or agrobacterium tumefaction. So if you follow my plant biotech class in the future, you will be exposed to these types of technique. So a cell that receive the gene of interest can then be recovered into the whole plant using a combination of plant hormone, and the whole combinations of the whole plant that are recovered can then be used to to experimentally determine the gene's functions or to enhance the crop potential for modern agriculture. So this is the downstream research example on using the plant oil cell. So in this research, we are looking or we are studying the functions of mechanism of the nutrient acquisition in the plant, in the oil plant. So we use several same types of decalers to discover of these poorly understood potassium transported gene functions. So I think that's all for today's lesson. I hope you enjoy it, and see you guys on the next lesson. Bye.